Hello lovelies, in this video I'm going to be taking you through your Edexcel Combined Science Paper 1, the predicted paper that we've written for 2024.
The top layer of your skin cells constantly shed as new cells from below grow and replace them. Describe the process that produces new skin cells. So this is a describe, and it's describe the process. So it wants the steps in the right order, and that order can sometimes be really important. So we want to make sure we can plan our answers so we're not going to get muddled up at any point. And obviously it's giving us some context about new skin cells replacing old skin cells. So these are going to be identical replacement cells, which means that the process that produces these new cells must be mitosis. So it hasn't mentioned this anywhere in the question, so we're probably going to get a mark for talking about and just mentioning the word mitosis. So we need to make sure we say that. And then I would literally just do this and write yourself a little plan of the stages of mitosis, write it out briefly, just so you know that you're happy with the order before we write our full answer. So the DNA is replicated, which is the first step. The chromosomes then need to kind of line up, meet in the middle, and then get pulled to the ends of the cell. And we say that these are the opposite poles or opposite ends of the cell. And then cytokinesis happens. And cytokinesis, if you can't remember that word, you can just say it's where the cytoplasm and cell membrane divide or split. You need to say both of those things though. If you are going to not remember cytokinesis, you need to say cytoplasm and cell membrane divide. And with all mitosis questions, whether they're long answer or short answer, we should try and always make sure we end with what gets formed. So two genetically identical daughter cells. That phrase should always be something that we end or add to mitosis questions, because that's what's going to give us the marks. Okay, so I'm going to type out my answer just so it's a little bit clearer for you, because it's going to be quite a long one. So the cells are produced by mitosis, I've got that word in there. The DNA is replicated, and then the chromosomes are pulled to the opposite poles of the cell. You can say ends of the cell, that's fine. The cell membrane and the cytoplasm then divide to form two genetically identical daughter cells. So I've formed this as 3 sentences, but really this is 4 marks. So 1 mark for saying mitosis, 1 mark for saying the DNA gets replicated, 1 mark for the chromosomes being pulled to the opposite ends of the cell, 1 mark for the cell membrane and cytoplasm dividing. You could have just said cytokinesis happens. And then also you can get a mark for saying the 2 genetically identical. So we've written more here than we need to, which is good. And this is just all the detail I know about the stages of mitosis, and it's in the right order, importantly. Doesn't matter if you miss out certain parts of this. So if you said the DNA was replicated and then you jumped straight to cytokinesis, you can still get marks because it's in the right order. As long as you didn't say some of the stages out of order or in the wrong place, then that's what would not get you the marks. The chart shows the stages of the cell cycle that skin cells go through. So this is clearly showing us that this is a cell cycle question. A skin cell typically takes 48 hours in total. Describe one change that occurs in the cell during stage 1 of the cycle. Okay, so this is a describe question, so it wants us to describe one change. So give some detail about a change that occurs in the cell during stage 1. So stage 1 is this large section here on the diagram. So if we think about labeling this so we know what the stages are representing in each stage. So the first stage, the one that's the largest amount of time, is going to be interphase. Then we've got mitosis, and then we've got cytokinesis. So even if you don't know these terms, so interphase, and then you can't identify those, that large stage that happens before cell division, we should know that that stage is kind of the growth and preparation stage before we go into mitosis. So what happens in this stage? So let's list some things that we could talk about. So in interphase, in this stage 1 before mitosis, the DNA is going to replicate, and we obviously have already talked about that in our previous question. The subcellular structures, so the organelles like mitochondria and ribosomes, start to replicate or increase in number. The cell increases in size, so it gets larger. And protein synthesis is going to happen because we're going to need to make a lot of different proteins in order for us to carry out various processes like DNA replication and mitosis. And we need to make sure that we've got enough DNA, 2 exact copies of the DNA, and enough organelles, so structures inside the cell, so that when the cell splits, both cells get enough of all of the subcellular structures and also get a copy of the DNA each. So I'm going to pick DNA replicates as my answer, but you can have any of these options. So I've just pulled the diagram over to this one so that we can look at it for part C, because we're going to need to use it. We need to calculate the length of time a skin cell spends in stage 2 of the cycle. Give your answer to 2 significant figures. Okay, so this is just a maths question. It's a common maths question around the cell cycle. There's normally going to be some kind of maths you have to do. So we just need to figure out how much time is taken up by stage 2, which is 1 out of these 10 sections. So this circle has been split into 10 segments. We just need to find out how long one segment is worth. And it tells us that the whole thing, all 10 sections, take 48 hours in total. So 10 sections takes 48 hours. We just need to find out the time taken for one of those 10 sections. You're going to get different versions and questions of like this. So you could get one where they actually put percentages on for you. You could get one with numbers on or times on. This is one of the ones that is sort of a little bit more abstract because it's just a circle cut into sections with no numbers and you have to kind of work it out. So different types of this question are going to come up. But this is how we would work out this one. It's simpler than you think. So first of all, we just need to find out how long 1 out of 10 sections is going to take in terms of time. Then we're going to have to convert because as you can see down the bottom where it wants our answer, it wants it in minutes. And obviously at the moment we've got 48 hours. So we are going to have to do a time conversion. And then it also wants us to make sure our answer is in 2 significant figures. So these are our 3 steps that we have to go through for 3 marks. So to find out how long 1 out of 10 sections takes, then we're just going to do 48 times 1 over 10 or divide 48 by 10, which gives us 4.8 hours. Then to convert that into minutes, we need 4.8 lots of 60 minutes because there's 60 minutes in an hour. So that gives us 288 minutes. And now having to put that into 2 significant figures, which is 290 minutes. So if you're not sure, to get from 288, we know we have to round up because it's an 8, so it's above 5. If it was below 5, then you would round down. But we're going to round up and it's going to be to 290 then because the 0 on the end of 290 doesn't count as a significant figure, so therefore we've got to 2 significant figures. Lots of people lose marks in the exam for not remembering to go back and check to see if it wanted it in a certain decimal place or significant figures, and it's easy marks to get. So just make sure when you've done a calculation question, go back and read the question again and just check to see if it wanted it in 2 significant figures, because it's easy just to forget that little sentence underneath the actual question and just make sure that you've got it right. Sometimes problems with the cell cycle can lead to cancer. So this question is obviously signposting then that this question is about cancer. So first of all, it wants us to define cancer. This is something we just have to know. Every time you get a define question, it will just be you writing out a definition that you have learned. So hopefully we have learned the definition for cancer. So it's uncontrollable cell division that causes the formation of tumours. If you just put uncontrollable cell division, that's enough to get you the marks. That's the important part that they want as part of the definition. And then for part 2, it's asking us to state a risk factor for developing skin cancer. So a risk factor is something that increases your chances of getting a certain disease in this context. And specifically, it put skin cancer. It didn't just say cancer, it said skin cancer. So we need to have to try and think about what of the factors that we know could increase our chance of getting cancer could be most related to or most likely to cause skin cancer. So we should have learned about some risk factors that increase your chance of getting cancer. The main ones obviously smoking, also exposure to radiation, ionizing radiation specifically, and that's our link to physics. Obesity can also be an increased risk factor for cancer, and also obviously large alcohol intake or prolonged alcohol intake. So if we were going to pick which one of these is most likely to develop skin cancer, technically all of these can be risk factors for cancer. But often things like smoking obviously is most closely related with lung cancer. Alcohol intake is most closely related with liver cancer. Obesity is normally around the internal organs. But we should hopefully know and have seen examples that obviously exposure to UV radiation such as sunbeds or getting sunburn and not using sun cream increases your risk of getting skin cancer because the UV rays are hitting your skin all the time. So I'm going to put exposure to UV rays or UV radiation. So although this pulls on your knowledge of risk factors for cancer, just need to do that little bit extra work to try and make sure our answer is relative to skin cancer. There are some other radiation, ionizing radiation examples you could put, which would also be correct. And these are options you can, but hopefully you've learned the example of kind of sunbeds or tanning, and you know that it's the UV rays that are more likely to cause someone to have skin cancer. Variation can influence the phenotype of an organism. Put a tick for each row to show what type of variation causes the following phenotype differences. So we've got to decide if each of these is caused by just genetics, by just the environment, or by both genes and the environment. So if we go through these one by one, blood type is definitely caused by genes. You inherit that from your parents. Height is both genes and the environment because it can be affected by your diet as well as genes. Genes coding for flower colour because again that's kind of like eye colour and hair colour in humans. And then the language you speak is definitely just due to the environment that you're raised in, where in the world you are raised. That is determined if you speak a certain language. It's nothing to do with genes. Genetic variation in a population is due to mutation. Define mutation. So this is just a definition. We have to know just how to define a mutation. So it's a change to the DNA. You've got some alternative wording in the mark scheme you can use as well. Do all mutations have negative effects? Explain your answer. So by negative effects, it means are all mutations bad for an organism and do they all reduce survival? So the answer is no, but we have to explain our answer. So we're not going to get a mark for just saying no. We need to give reasons why there are not all negative mutations. Why are there some mutations that are positive, for example? So we're going to say no because some are neutral and some could be positive, but we need to maybe give a why, so like a reason why they might be positive. So if you think about the fact that we've said that negative means it reduces an organism's survival, positive does the opposite, so it will improve or increase an organism's chance of survival. And then it's always good to give something like an example with these. As you can see from the mark scheme, if you just put because some are neutral and some are positive and therefore they can help survival, that probably would get you the 2 marks. But if you just said one or the other, you would need to give an example in order to kind of illustrate your point and explain your point. So I'm going to use an example of antibiotic resistance. There's an example in the mark scheme about disease resistance, which is a similar thing, something that could help an organism to survive. So we're going to say no, but remember that doesn't get us a mark. So no, not all mutations have negative effects because some mutations can be neutral. And then you could give an example, but I didn't because I also said they have no effect on survival. And some could be positive because they increase the chance of survival. For example, antibiotic resistance. Communicable diseases can be caused by a virus. Describe what is meant by the term communicable disease. So hopefully communicable disease is a term that we know, and we know that the communicable type of diseases are the diseases that can spread from one person to another. And if you're going to describe it, we're not explaining how they spread or anything like that. We're just literally giving detail about what a communicable disease is. So all we need to say is our definition, which is going to be that it's a disease that can spread from one organism to another. You can put between people, but obviously we know that not all pathogens and not all communicable diseases are from human to human. Some are between animals, some are between plants, some are between humans and animals. So organism to one organism to another organism is a better general way of describing it. Describe how the human body prevents the entry of pathogens that cause disease. So again, we have another describe, and this time to describe how, which normally talks about kind of processes, kind of details kind of how something works. And in this case, it's asking us how the human body is going to prevent the entry of pathogens. That means how does the body stop pathogens from getting in? So this is about the body's non-specific physical and chemical defences, not the immune system, not what happens once the pathogen gets inside the body and how the body fights off. It's about how do we actually stop pathogens getting in the first place. So this is a 6 marker and they are normally levelled answers, so we need to give detail and logical linking. So sometimes this is about doing it in the right order, sometimes it's just about the level of detail, and in this case we'll kind of maybe be linking some things together. So the main 3 things we should obviously know about how we stop pathogens getting to the body are the skin, which acts as a barrier, and also obviously there's clots there, which is part of the blood job to help prevent pathogens getting into the body. You've got your mucus and your cilia, which line your airways, so your nose and your throat, down into your trachea. They obviously produce the mucus, which traps the pathogens, and then the cilia move the mucus with the trapped pathogens up and out to get rid of it out of your body. And the stomach contains the acid, which we know kills bacteria in food. So here's my answer. I've got those three different sections explained in detail and the organs where this is going to be happening. So the skin acts as a physical barrier to prevent entry of pathogens to the body. If there is a cut, then platelets in the blood form clots, which seal the wound with a scab that prevents pathogen entry and infection. So this is obviously my first paragraph about kind of one section of the three that I'm going to talk about. I've mentioned the skin, I've mentioned the blood. I've also linked here, so I've linked the two together, so how if the skin acts as a barrier, but if there's a break in the skin, then this other thing steps in to help us, which is the blood clots. I've also really kind of signposted pathogen entry as well. So at the nose and in the trachea, the cells lining these airways produce sticky mucus, which traps pathogens. There are also cilia on cells which move mucus to the throat to be removed through coughing, sneezing, or being swallowed. This then links to the next paragraph, so that's why I put this one first. The stomach contains hydrochloric acid, which kills pathogens such as bacteria in food or in mucus that is swallowed due to its low pH. So I've highlighted here where in every paragraph I've tried to make sure I'm showing that I'm linking back to the question, that what I'm saying is actually answering the question, because I'm talking about preventing the entry of pathogens, preventing pathogen entry, trapping pathogens, and then removing them, killing pathogens as well. So making sure that each one we're going to talk about, you're showing the examiner that you understand what you're answering. There was another section in the mark scheme about eyes, because obviously you've got tears, and the tears contain enzymes, or they have a low pH, which makes it unsuitable for bacteria. Also, we didn't mention that there are kind of oils and things on your skin that prevent any pathogens from growing on your skin as well. But that you don't have to have every single bit of that detail in there to get a level 6 answer. So we had to mention 3 or 4 different kind of organs or places in the body, which we've done with detail, correct detail, which we've done, and then some logical linking between some of the points, which we've got as well. So it's not just a bullet point list, okay? Because sometimes, especially with these levelled answers, a bullet point list might not get you the full marks. HIV is a virus that can result in the condition known as AIDS if left untreated. Name the type of drugs that can be used to treat HIV infections to prevent AIDS developing. So this question is asking for the type of drug that is going to specifically be used to treat HIV infections, not just viral infections. And when it says type of drugs, it means things like antibiotics or painkillers or types of drugs we've learned about. So there is and only should be one type of drug we've learned about that can treat HIV, and that is antiretroviral drugs. If you put antiviral drugs, then you don't get the mark, because that would not treat HIV specifically. That's just used to treat viruses. Patients diagnosed with AIDS usually die from a different type of infection, such as a bacterial infection. Explain why. So we need to give reasons why other infections might kill a patient who has AIDS. So this is just about us being able to explain or use our knowledge of AIDS to explain why this happens. So we need to be able to make sure we understand what AIDS is as a disease and how we can link it to this idea that AIDS isn't the thing that specifically causes the death of people with this disease, but other infections can. So AIDS is a disease where the white blood cells have been damaged to the point that the immune system is too weak to fight infections. So if you have AIDS, the HIV infection that you've had has damaged your white blood cells so much, killed so many white blood cells, that you now basically have a non-functioning immune system. So there are no white blood cells, or very, very few white blood cells. And so why would another infection then be able to take hold and potentially kill you? Because you don't have enough white blood cells to kill the pathogens. So normally your phagocytes or the B cells will be producing antibodies, and all of this would help get rid of and kill pathogens in the body if you have an infection, but they can't do that because there's not enough of them if you have AIDS. So then the bacteria can replicate and cause an infection that can be serious enough to kill you. So these are our 2 marks then, talking about the fact that we don't have enough white blood cells because of the AIDS, and why then that leads to a pathogen being able to infect someone is because there are no white blood cells in order to kill it. So AIDS patients have a severely damaged immune system, so there are not enough white blood cells to kill bacteria effectively. And there's variations on this in the mark schemes that you can use, but as long as you've got that idea of having a damaged immune system, having no immune system, not having enough white blood cells, and then therefore the bacteria cannot be killed, or pathogens can't be killed effectively, as they usually would. The graph shows the number of new HIV diagnoses each year in England since 2013. Describe the pattern the graph shows. So when it comes to describing graphs or tables, it means we've got to say what we see. We're literally telling them what we can see in terms of the pattern in the graph or the table. In this case, it's a graph, so we're going to literally say, is it increasing, is it decreasing, when is it increasing, when is it decreasing? There's 3 points here, so we've got to say 3 things. Often we would talk about kind of the general overview of a graph, if it's a consistent pattern, but if it changes midway through, you talk about the 2 different sort of patterns you can see, an increase and then a decrease, for example. And then we need to give some data points. So there's normally always marks for using data as reference to back up your descriptions. So first of all, let's start at the beginning. So between 2013 and 2014, we had an increase. And then after that, we had a pretty steady decrease from 2014 to 2022. And that decreased from about 2,500 all the way down to about 700. So this is 2 things we can say. We can talk about the increase, then we can talk about the decrease, but we need to put at least one data point in there, like from 2013 to 2014 for the increase to get that extra point. So there was an increase in the number of new HIV cases from 2013 to 2014. So that's my first point about the increase, plus I've added the data point. Then the case numbers steadily decreased from about 2,500 to 700 in 2022. So I've then talked about the decrease and I've added some numbers on there as well. So some common mistakes with answering questions like this is that people try to explain why the graph could be decreasing and not describing when they think that they can only say one thing and they're not sure how to get 3 marks. But that wouldn't get you any marks here, talking about why. Not using data, so again, sort of just saying, oh, it decreased overall, there was an increase at the start and then it decreased, or it rose and then it fell, but not giving any data to sort of support their points. Part 2, suggest 2 reasons that could explain the change in the number of people diagnosed with HIV since 2014. So 2 is involved, so we need to give 2 reasons for 2 marks. And it wants us to think about what we've just looked at and how we've talked about the change. We know what the change is, that's a decrease, because we can see that on the graph and we've also commented that it was a decrease from 2014. So why? We need to come up with some reasons why this might have happened. So this is a suggest question, which means we are using our own knowledge and information from the question, right? So it's not just something you will have definitely learned, but you're kind of having to interpret from the question and maybe use a bit of common sense or something that you've learned from your scientific knowledge. So if there's been a decrease, less people are being infected with HIV since 2014 every year. Why? This is what we've got to try and give explanations for. So in terms of the knowledge that we need to use for this, if we know the stuff that we have learned, the content knowledge we have learned will be about how the spread of HIV can be prevented. Because clearly the spread is being prevented from person to person because there's fewer and fewer people contracting the disease every year. So we know from our content knowledge that condoms help prevent the spread of HIV. We know, and we've already answered a question about this, that antiretroviral drugs can help prevent the spread as well. Now, because this isn't just a content question, we can't just say these things. We should turn these things into what could have changed over time to help reduce the spread. So you can't just put condoms as a reason because they have been around for a very long time. So in terms of what difference, what change could there have been since 2014 that might have made this decrease more likely, then we could say something like condoms being more widely available or cheaper or even freely available, potentially. We can talk about the antiretroviral drugs being developed to be better. There's new drugs or different drugs or better drugs being developed because, again, the drugs have existed for a while. And then we can think of some things that maybe aren't directly linked to the content that we've thought, but things about awareness or education. So people know more about HIV, know more about how it's spread, and know how to protect themselves from getting it, i.e. using condoms, and that can help prevent the spread. Or more testing and screening. So if people know and are aware that they have HIV earlier on, then they're less likely to be able to pass it on to other people because they can carry out the steps they need to stop spreading it. So all of these are things that we can put to answer this. We only need to pick 2. But you can see how there's a difference between the 2 things, sort of statements of fact that we would have learned from content, and then turning that into kind of an analysis of what things might have changed or what improvements might have been made in order to prevent the spread of a disease. And this is a common type of question around diseases in this topic. So be prepared to be able to do this. And things like awareness, education, more testing, more screening, all of that stuff, and some of the other things mentioned in the mark scheme as well. These are all kind of common answers to these questions. They can be used for any disease, not just HIV. So I've put improvements in antiretroviral drugs and more people being tested for HIV. A group of students investigated the digestion of fats in milk by the enzyme lipase. So the first question answers to identify which of the following diagrams best represents a fat molecule, and we're only allowed to tick one box. So another word for a fat molecule would be a lipid or a triglyceride. You may have heard these terms as well being used. So we tend to use kind of fats and describing fats when we are talking about stuff at key stage 3, and now you probably hopefully heard them used as a lipid or described as a triglyceride at key stage 4. So we should hopefully know that these molecules, lipids or triglycerides, are made up of one glycerol molecule and 3 fatty acid chains, because we know that that's what happens when we break down lipids, or when the enzyme lipase breaks down lipids. So out of these 3 molecules, which one looks like it's made of a glycerol and 3 fatty acid chains? So it's got to be the middle one, and hopefully you'll have seen them maybe look like this in diagrams. The other 2 can't be the right answer because they're both polymers. So they're chains of sugars or chains of amino acids for proteins, which create these long chains of same molecules joined together. But lipids are different, and so that's why it's the middle option. State where in the body the enzyme lipase usually works to carry out fat digestion. So this is now getting obviously into digestion content. So this is asking us where in the body, so where in the digestive system will the enzyme lipase work? So be careful, it's asking where it's working, not where it's being made. So hopefully we should know that digestion occurs in the mouth, the stomach and the small intestine. In the mouth, it's only amylase that works in there. In the stomach, it's only the protease enzymes that work in there, whereas the small intestine, all 3 of them work in there. So the amylase, the protease and the lipase. So in terms of where the lipase actually works, it's only the small intestine. And we have to make sure we put the small intestine, you can't just put intestine. The student sets up their equipment as shown in the diagram. A small beaker containing milk at 10 degrees C was set up. Lipase solution was added to the beaker. The students measured the pH of the reaction mixture using a pH probe attached to a pH meter. The pH was measured again after 30 minutes. They repeated the experiment at the following temperatures: 20, 30, 40, 50 and 60 degrees. So if we're measuring the pH, that is going to be our dependent variable, and this is what I would do when presented with lots of text about an experiment. Then make sure as you go through you're kind of highlighting and annotating this stuff. So what can you pull out, what can you identify from the information they've given you, and then we can use that to help us answer the question. So they measured the pH of the reaction and then they repeated the experiment, but they changed the temperature each time. So that's going to be our independent variable, so we change temperature. And just remembering and making sure we understand and remember how we identify each one. So the dependent variable is always what you're measuring and the independent variable is always what you're changing. So luckily the question is asking us to give the independent and dependent variables, so now we can answer that really easily. And I would do this even if that wasn't the first question, because then I'd know and have clear in my mind which one was which, because there's lots of different questions that they could ask that this could be useful for. So dependent is measure and independent is change, DMIC. Some people remember this by remembering the phrase dairy milk ice cream. So our independent is temperature and our dependent is pH. Name the piece of equipment the students could have used to keep the beakers of reaction mixture at the correct temperature for 30 minutes. So this is just one of those ones that, again, we just have to know our pieces of equipment and what we can use. So hopefully you've done an experiment at some point where you've had to keep something at the same temperature for a long time and you've used a water bath. A lot of the common answers to this question would be the thermometer, and people think because it's there in the diagram it's a clue or a hint, but actually the thermometer measures temperature. It's not going to be able to control it or keep it at a set temperature. The students' results are shown in the table below. So we've got the temperature and sort of all the different temperatures they tried at the pH that was measured at the start, and then the pH of the lipase and the milk mixture after 30 minutes. Explain why the pH decreased when the lipase solution was added to the milk. So because it's an explain question, we just need 1 mark and the reason why the pH decreased when the lipase solution was added to the milk. So if we remember, at the start of the question told us that the students were investigating the digestion of fats in milk. So when you add lipids, so the fats, and the lipase enzyme, what do you get? Well, the lipase will break down the lipids into glycerol and fatty acids, as we said at the start of the question. And if we noticed, we've just written the word acid. So fatty acids are acids, that's why they're called fatty acids. So if you're producing an acid, the more acids you make, the lower the pH is going to be. So the reason why is simply because fatty acids are produced when the lipase breaks down the lipids. Using information from the table, first explain the effect of temperature on the activity of the lipase enzyme from 10 to 40 degrees. So it only wants us to focus on 10 to 40 degrees, where we can see we obviously get a decrease at the start of 5.2 at 10 degrees, and by the time we get to 40 degrees, it's decreased to 2.8. So we get a lower pH with higher temperatures. So it's in the same amount of time, so it's still in 30 minutes, which means that we're actually getting a faster rate of reaction as the temperature increases. So really this question is asking you to explain why does increasing the temperature increase the rate of reaction. So hopefully we're happy that this is a chemistry question. It's effectively asking us why increasing the temperature increases the rate of reaction. Well, hopefully we should know that it increases the movement of the particles, so the enzyme and the substrate are going to collide more. This is the same answer you'd give to any increased rate of reaction question that's asking you to explain it based on an increase in temperature. So increasing the temperature increases the rate of reaction. That's what we've got from the table. This is because at higher temperatures, particles are moving faster, so there are more collisions. And that's as much detail as you need to give. You can say the enzyme and the substrate collide more, that's also fine. So I've given you kind of both alternatives here. But this is one of those examples where it's chemistry knowledge. We will have learned about this as well. We've been done enzymes in these biology lessons, but this is also kind of a crossover question as well, a little bit, taking some of your chemistry rate of reaction knowledge. So now it wants us to explain the results when the temperature was increased above 40 degrees. So if we just look at 50 and 60 degrees, at 60 degrees there is no change in pH after 30 minutes, and at 50 degrees there's very little change in pH after 30 minutes compared to the changes we saw from 10 to 40 degrees. So hopefully we've sort of can recognise that an increase above 40 degrees is going above the optimum for body temperature, which is where these enzymes normally function. And so anything above the optimum where we lose or see a drop or decrease in the rate of reaction is because of a denatured enzyme. So the denatured part here, that key word we need to mention, but because it's an explain question, we also need to explain why that denaturing leads to that decreased rate of reaction. So the reason why denaturing causes a drop in the rate of reaction is because the active site changes shape so that it can no longer bind to the substrate, and that's why we don't get a reaction anymore. So my first point is to say that above 40 degrees, the rate of reaction slows then stops because the enzyme has been denatured. Now that first part about it slowing or stopping doesn't necessarily need to be in your answer, but it didn't say decrease, and so it's always worth mentioning it if you have identified the pattern. So that's our first point, and then we need to explain why being denatured means that it can't carry on. It's so that it can no longer bind to the substrate. Common mistakes with questions like this: not using keywords, so not saying denatured, not saying enzyme, not saying active site, not saying substrate, just missing out those words and kind of being too general in what you're saying. And then not fully explaining why. You might get 1 mark for saying, oh, because the enzyme denatured, full stop. But that's only going to get you 1 out of 2. Not fully explaining it and talking about the fact that it can no longer bind to the substrate is why people lose marks here. The diagram shows the evolutionary tree of part of the big cat family, Felidae. Wants us to state which big cat is most closely related to the jaguar. On the family trees like this, the most closely related ancestor will share a branch or be on the closest branch to the one you're being asked about. So in this case, it shares a branch with the lion, so you can see they've got a join there, so that's our answer. Now it wants us to state the genus of the clouded leopard. The genus is always the first part of the binomial name because we use the genus and species taxonomy groups to make the binomial name. So Neophelis is the first part of the binomial name for the clouded leopard. And then lastly, state the species name of the snow leopard. So the species name is the binomial name. So we need both parts of the name. We don't just say the second part. It doesn't make any sense without the genus. That's why we have the binomial name. It has to come in 2 bits. So if we're doing the snow leopard's binomial name or species name, it's Panthera uncea. Common mistakes made with these questions is being careful when copying. You are copying down Latin names, so be careful with spelling, but also make sure you use capital letters and small letters where you should as well. And remembering that the species name is the binomial name, so you need both parts. The snow leopard is quite different from the other cats. who lives in very cold, snowy, mountainous habitats. Suggest a feature of the snow leopard that helps it to survive in its environment. So this is an adaptations question. That's what it's really asking here, about features that help an organism to survive. So it's also a suggest question. So we need to use our knowledge about adaptations and also the question information which tells us it's very cold, snowy, mountain habitat. So we need to give specific adaptations to this information about being very cold. So adaptations we'd expect to know or have learned for cold environments would be something like a fat layer to insulate, thick layer of fur or hair, or having a small surface area to volume ratio to try and reduce heat loss. So I'm going to go with a thick coat of fur to reduce heat loss. We need to make sure we add how it helps it survive. So it's only 1 mark, but you can't, I'd rather than just put a thick coat of fur, making sure you've added that extra bit so you're linking it directly to why it's an adaptation to the cold and how it helps it to survive. Unlike all other cat species in the same genus, the snow leopard cannot roar. There is a debate about whether it should have its classification changed. So this would mean it would not be in the same genus anymore as the others. Describe a method scientists could use to determine if the snow leopard is significantly different enough from the other cats to be put in a different genus. So when it asks to describe a method, it's talking about what are the steps in the process and how would we be able to tell or decide if something is significantly different in terms of relatedness from other organisms. So normally we compare and talk about similarities in their DNA. That's how we make these phylogenetic trees. And how do we look for differences or similarities? We compare the base sequences. So we would need to sequence the DNA of the cats in the genus, so all the cat species in the genus, and the snow leopard as well. And then we would need to compare the base sequence of their DNA to see how different they were. Variation may arise due to the geographic isolation of a species. Explain how geographic isolation of members of one species could lead to a new species evolving. So by variation, they mean differences between organisms. Geographic isolation means being separated in space from each other. So either side of a mountain, either side of a river, far apart, high up a mountain, down the bottom of the mountain. These are all the kind of examples of geographic isolation. And then it's asking us to explain how it happens. So reasons that link this idea of geographic isolation to a new species and how that process actually comes about forming it. So hopefully a new species evolving is something that we know, and we know this as speciation. So the process is speciation, and we've got this idea of like there's two populations of a species, A and B. They're one species, but they got separated geographically for some reason. And so either side of this, in these two new environments, we're going to have different environmental conditions. And then natural selection is going to take place in either side of the new environments. And if we're forming a new species, then it means they must not be able to breed with each other anymore. So that is enough really for 3 marks. This isn't a really long answer question. We're not having to go through the whole process of like evolution. We're just giving an overview for 3 marks. So we're going to mention the process of speciation because that hopefully will get us a mark. Then we're going to say that the different areas are going to have different environmental conditions. We're going to briefly describe what natural selection occurs so that only the best adapted members in each environment survive and breed. And then what makes them new species is eventually the two populations can no longer interbreed, so they are new species now. Now I've probably said more than enough for 3 marks, but this is still a decent level of detail and kind of what you need to be talking about if you're talking about speciation. Describe how the powdery mildew fungus can spread from plant to plant and how it enters the leaves. So it's a 3 mark question. First of all, we want to tell us how does it get from one plant to another. So how does it spread? And then also how does it get into plant as well? How does it get inside? So we should know that fungal diseases, regardless of whether they're plant or human ones, they produce spores and they are spread by the wind and the rain from one plant to another. And then it says, how does it enter the leaves? So there are holes in the leaf, which are stomata. So this is an obvious and easy place for spores to get in. They're very tiny. And even if it's growing on the leaf, it can kind of grow around and grow inside. So it's a really easy entry point. So the fungus produces spores, that's going to get us 1 mark. which is spread by the wind, going to get us the second mark, and they can enter the leaves through the stomata, the third mark. The infection will eventually kill the plant because it is no longer able to produce chlorophyll. Explain why the plant dies. So we need reasons why the plant dies, and we've got to link it to it not being able to produce chlorophyll. So it's given us that bit of information at the start. So if a plant doesn't have chlorophyll, then it's not going to be able to carry out photosynthesis. That still doesn't tell us why the plant dies. We need a bit more information. So why does the plant die if it can't do photosynthesis? Because it's not able to produce sugars. Linking that to why no sugars would mean the plant dies because it can't make new molecules, like proteins, for example, or release energy. So it's not going to have enough energy or molecules to be able to carry out its living processes or grow or make new cells or do anything because it doesn't have food effectively. We also need to think about, we can't just say no chlorophyll, it means no photosynthesis. Why? Because chlorophyll is where the light energy is absorbed for photosynthesis. So without it, without that green pigment, then it can't absorb light energy for photosynthesis. And then we can say if we can't make photosynthesis, then we can't produce sugars. And then we can link that to a reason why the plant would die, because that would give us our 3 marks. So without chlorophyll, the plant cannot absorb light energy, so no photosynthesis can take place. This means the plants can't produce sugars for growth, and that's enough. You could say sugars to release energy for growth, or sugars to make new molecules for growth, but just saying for growth here in this context is fine. If it asked why the plant doesn't grow, we would have to give a bit more detail. We couldn't just say it can't grow because it doesn't have sugars. You'd need to give a bit more information. Scientists are investigating ways to genetically engineer barley plants to produce similar antimicrobial substances. Explain how scientists could use a gene from garlic to genetically engineer barley plants to produce the same antimicrobial substances. So explain how would be the steps in the process that they would use to make this happen. It's about genetic engineering, which is the transfer of genes from one organism to another. In this case, we're transferring the gene from garlic to barley, and we would need to cut out the gene for the antimicrobial substances from the garlic and then transfer it into the barley. So this is testing us on our ability to remember the stages of genetic engineering. So we're going to need to cut the gene out, and we're going to need to mention restriction enzymes in order to do that properly. This will create sticky ends. We should mention these. Using the plasmid as a vector is another thing we should mention, and then the ligase enzyme that we need to join the DNA into the plasmid. And then we've got to obviously transfer it into the plant, but we would need to do that at an early stage of development if we wanted the whole barley plant to be able to make these substances and not just a few cells. Scientists would need to cut the gene that codes for the antimicrobial substance out of the garlic DNA using restriction enzymes. A plasmid vector would also be cut using the same restriction enzymes so that they had complementary sticky ends. The garlic gene and the plasmid DNA can then be joined using the enzyme ligase. The garlic gene can then be transferred into the barley cells during an early stage of development to ensure the plant can make the substances all the way through. So we need to make sure, obviously, we're putting in all of those key points, which I've done here to get the 4 marks. So although I've written them in full sentences, you can see from the mark scheme that the kind of really key points you need are cutting the gene out using restriction enzymes, talking about a plasmid vector, and talking about using the same restriction enzymes to get sticky ends so that they match, and then joining the DNA and the plasmid vector using the enzyme ligase, and then ideally that would get us 4 marks. We also obviously need to mention the early development when you're transferring it. So if you're doing a genetic engineering question, you have to talk about the transfer, where's it going from and to, and if it's plants, we need to make sure we're doing it at an early stage of the development.
Well done for making it to the end of the paper. This was quite a long one, so you definitely deserve to go and have a rest now.